we will have Dr. Ty Pettay. Dr. Pettay has a BS in Biological Sciences from Clemson University, and then a master's in Marine Biology from College of Charleston, and he's followed up with a PhD in biology from Pennsylvania State University. I know Ty personally just from working with him through the foundation, working on projects, grants, and he is helping me with our citizen science program. So I think everyone will really enjoy today's talk, as I enjoy listening to Ty talk myself. So without further ado, I think Ty, you can start if you want to log on and share your screen.
Great. What are you going to say?
No. Go ahead.
Thanks, Chris. I appreciate it. Thank you for the introduction, and thank you for inviting me to give this talk. My talk that I'm going to be giving today, as I try to share my screen so you can see that okay?
Yep, looks good.
Is going to be about work that I did primarily during my PhD research. But hopefully if I don't ramble on and run too long, at the very end, I'll turn this back around and talk about what it has to do with work I'm doing in the Port Royal Sound now. And so much of the work, or all the work I did during my PhD and my first postdoc was related to coral reefs. And so I'm talking about tropical coral reefs today. But there's a lot of connections to the work that I am doing in the Port Royal Sound and trying to get started in the Port Royal Sound now. Before I get going, I just wanted to most of this research was sponsored by particularly the National Science Foundation, and then some smaller pools of money from Florida International University and then Penn State. I won't dwell on this slide too long. I was going to give you a quick background on myself and my research interests and the kind of research I do. Chris already went through this list pretty well. The one place he didn't mention was Florida International University. So I started my PhD down in Miami and at FIU, and then our lab moved to Pennsylvania, or to Penn State, and that's where I finished, got my PhD. After that, I went on and did 2 postdoctoral stints at the University of Delaware at their marine campus in Southern Delaware, and also had a short stint as a research faculty scientist there before I took the job here at USCB in Beaufort. So you can see I'm originally from South Carolina, and was very keen to get back to the Low Country and to the state to do research. And so I'm excited to be back at the USCB and doing local research that matters to our state and particularly the Low Country. So again, my research and my lab really use a combination of research techniques that incorporate molecular genetics, comparative physiology, and environmental monitoring. And I use these techniques to answer kind of 2 broad questions. First being, how do microalgal populations, microalgae, and we'll talk more about these in the coming slides, are essentially single-celled plants, and in my case, live in the marine environment. But how do these single-celled plants, their populations, species, and communities respond to environmental change? And then conversely, it turns out that these particular organisms can influence the environment and biogeochemical processes in coastal systems too. And so I'm interested in how, as these single-cell plants respond to the environment, how do they influence other processes occurring within coastal systems like coral reefs, but also like estuaries like the Port Royal Sound. And so I'm going to touch on all 3 of these techniques and these 2 questions today, but most of the the work I'm going to talk about is really more the molecular genetics and answering this first question, or not answering it, but addressing it for one subset of species, how these microalgae respond to environmental change. So I asked these questions and used these techniques in three main research systems: coral algal symbioses, which I'll be talking about today, but then I'm also interested in harmful algae and estuarine phytoplankton. And you may be asking yourself, how in the world am I going to tie coral reef organisms into estuarine phytoplankton? As you'll see in the coming slides, coral is actually a symbiosis that includes single-celled plants or microalgae. And so we can ask very similar questions in these very different systems. And so I'm not going to read all these, but these are some of the topics that I am interested in, have researched and published on in the past. So for the talk today, first I want to introduce a little bit more about microalgae, what they are, how they function, their diversity, and then I'll go into a crash course in kind of coral reef biology, and particularly coral algal symbioses. So at the tail end, I could present a little bit of data that I gathered during my PhD dissertation. But I want to introduce microalgal diversity because you'll see in a minute, and as I just mentioned, a particular group of microalgae, specifically dinoflagellates. Hold on. Sorry, I'm trying to move my window in my corner. Dinoflagellates, which are right here. These are the group of microalgae that form symbioses with coral reef organisms. But so let's look at this weird looking tree over here on the right before we get to this image on the left. This is just a phylogeny, so it's like a family tree, but instead of having brothers, sisters, cousins, grandparents in it, it has different groups of single-celled plants and how closely or distantly they are related, and essentially how they evolved from common ancestors. So for those that don't know about phylogenetic trees, the branching points are where a common ancestor existed, and then for whatever reason, these two groups started to diverge, in this case, the dinoflagellates and the apicomplexans, and became their own separate, genetically distinct group. And so what this tree is really showing, it's a tree of life. It doesn't include the bacteria in this. This would be a separate tree, but this is all eukaryotes, which include us. And so all of these colored lineages that you see are different and distinct groups of microalgae, or these single-cell plants. And so you can see there's numerous different groups, and the length of these branches is genetically and evolutionarily how distant they are from their most closely related relative. So short branches mean they're more closely related, long branches mean they're more distantly related. But you can see, if nothing else, that there's numerous groups and they're quite divergent from each other, especially if you consider, say, us as an animal. Here's our little animal branch down here, very closely related to mushrooms, for example. So very little distance between these multicellular animals compared to the great evolutionary and genetic distance that you see between all of these microalgae. And so it's really quite a diverse group. And what's also interesting about this group is all these dark lines within the microalgae groups are actually non-photosynthetic. So that means they're not plant-like animal or plant-like organisms. They do not photosynthesize. And so this group contains not only photosynthetic, but non-photosynthetic organisms too. So it's a really incredible group. And so microalgae incorporate a wide diversity of organisms that are collectively commonly put into groups of protozoans or protists, which is just a hodgepodge of single-celled organisms, some closely related, some very distantly, if hardly related at all. So we have a large diversity of microalgae out there. genetically. So this tree is determined by gene sequences. And if you jump over here to the left, I like using this image because not only is there a huge diversity in the genomes between these, or divergence in the genomes between these organisms, but there's actually quite a large difference in size too. And so up top here, we have examples of various microalgae species, or in this case, genera, and their size. This will be your smallest Prochlorococcus, which is actually not on this tree over here, but is a cyanobacteria. So it's actually a photosynthetic bacteria. And these are actually the original single-celled plants that existed and gave rise to all of the photosynthetic organisms. But they're going to be the smallest. These organisms you can hardly see, if at all, see under a microscope. And then as you move to the right, you get increasingly larger and larger. And the sizes of these are in micrometers. So you're still looking at these micrometers, which is 10 to the -6 meter. You're still looking at these under a microscope to really see them. Although some of the bigger diatoms you can see with the naked eye or a less powerful microscope like a dissecting scope. But what this image is really showing is increasing size in microalgae and how, if it increases relative to things that we can see and touch in the real world every day. So if you compare Prochlorococcus, if it's relatively the size of a fish for things that we can see with the naked eye, the largest single cell microalgae is going to be the size of the Eiffel Tower or large buildings. So when you put it in that perspective, you can see there's quite a range in not only genome diversity, but also size in these microalgae. In the end, though, they are still small. They're called microalgae because they're, for all intents and purposes, microscopic. And so you need specialized equipment to be able to see and work with them. And so while these organisms may be small, they are in fact very mighty and very important to the world, and particularly the marine ecosystem. They're important for oxygen production, but also as a food source. And even though they are small and you need a microscope to see them individually, you can actually see them from space and under the right conditions. And so this is a diatom bloom here. Here's off the coast of New Jersey and New York. This kind of green, milky green kind of color swirling around here in the Atlantic Ocean is actually a bloom of billions of diatom cells that, for whatever reason, the right environmental conditions, temperature, light, nutrients, cause them to bloom and grow uncontrollably. And this is an image from space. And so you can see these microalgal blooms from space. So it's quite spectacular. And then, of course, since we're talking about coral reefs, I wanted to mention the Great Barrier Reef. And you'll learn more about this in a second. But the coral, the organism that forms reefs and formed the Great Barrier Reef, is able to do what it does because it forms a symbiotic association with dinoflagellates, like I mentioned before. And this is actually the largest biologically constructed system, it's not the right word, but biologically constructed system in the ocean, and you can actually see it from space too. This image is a little hard to see it because it's zoomed out so far, but if you just zoom in, you can see the Great Barrier Reef from space. So these very tiny organisms have a huge effect on global processes and ecosystems. And so then lastly, just to mention, while there's a diversity in genomes or genetic diversity and in size, there is also, this diversity has led to diverse physiologies too. And so you can find microalgae in just about any habitat and part of the globe that you look. So there are freshwater microalgae, there's marine microalgae. The green you see here are free-living phytoplankton. They can be planktonic, as I mentioned, floating in the water, or they can actually be living in the benthos in the water, so on the seafloor, for example. They're found in extreme environments like hot springs, or there's sea ice algae. They're found underneath ice sheets at the poles. And you can find them, as I mentioned, in the poles all the way down to the equator. And then lastly, not only are they typically aquatic, which you see in all these examples, but there are terrestrial algae too, or microalgae. And one of the best examples here is this green stuff you see growing on rocks and trees is actually another form of a symbiosis with microalgae and a fungus and what we know as lichens. So huge diversity in their genomes, but also physiological diversity too. All right, so our kind of last review slide for microalgae is going to be the dinoflagellates. And of course, I'm choosing this slide to focus in on because this is the microalgae we're going to focus on today with regards to coral reefs. So these are by far not the dominant phytoplankton or microalgae on the planet, or at least the oceans, I mean. That distinction goes to diatoms. Diatoms dominate the world's oceans, but dinoflagellates play an important role too. They tend to grow more slowly than other groups of microalgae like diatoms, but in conditions that diatoms are not favored, dinoflagellates can take over and do quite well. They of course form symbioses with coral reef invertebrates, as I mentioned, but some of them are bioluminescent, like you see here. So this dinoflagellate bloomed off the coast. I forget where this image was taken, somewhere in the Indo-Pacific, but that bloomed off the coast of this island. As the waves crash, the crashing waves irritate this dinoflagellate, and they actually have a protein luciferase that they release and modify, and it causes it to glow blue, kind of similar to what fireflies or lightning bugs do. But some will bioluminesce. Companies have even tried to make money off this. I don't think this actual toy exists anymore, but I think there's different versions of it where they would send you saltwater media to grow your bioluminescing dinoflagellate in, and then you shake it, put it in the window to let it photosynthesize, and then you shake it up at night and it would actually glow. So some bioluminesce, others can produce some quite nasty toxins. And so if you've heard of harmful algal blooms, many of the toxin producing harmful algal blooms, particularly in the marine environment, are your dinoflagellates. And so they produce some nice diseases like paralytic shellfish poisoning, diuretic shellfish poisoning, that can be quite harmful to humans and even wildlife. But not all dinoflagellates produce toxins, but some can. And they're called dinoflagellates because they have these flagella. If you can see this on your screen, they have one that trails from behind, kind of like a tail. Kind of see it on this image here too. And then they actually have this girdle around its waist almost, where another flagella will lay down and wrap around it. And these two flagella beating together can help it swim through the water column. I also like dinoflagellates just because they come in these really ornate and fancy forms too. They're quite amazing microalgae. All right, with a crash course in microalgae finished, let's get on to coral. And so a bulk of what I'm going to talk about is really not so much data that I have generated, but just kind of an overview of what coral are and what a coral reef is, why they're important, and then some of the stressors they face. And that'll give us a good segue into talking about a little bit of genetic data that I generated previously during my dissertation. So when I use this slide in teaching my students at USCB, I would generally ask people to yell out what is a coral, but since we're online, I'll answer the question myself. But this is a coral colony we have here. This is considered a branching colony because it has branches kind of like a tree would. And when you say what is a coral, the coral itself is actually an animal. It has a very thin, millimeters thick tissue layer that sits on top of a calcium carbonate skeleton, and I'll show you pictures of that in just a second. And then within the tissue of this coral animal we have these Dinoflagellate Symbionts, these microalgae that I was talking about. And so they actually live within the cells of the coral, so they're intracellular, which is by itself pretty amazing. They're able to get inside the host cell and evade immune responses. So they're either hiding in there or the coral is letting them survive. There's debate on what's actually going on there, but they live inside the coral tissues and multiply into really high densities. And then lastly, the coral itself also produces, and there's help from the symbiont through physiological processes, but they produce a calcium carbonate skeleton. So it's a hard part. And it's really this calcium carbonate skeleton that makes a coral reef a coral reef. So a little bit more about the symbiosis itself, and then we'll talk more about reefs in general. As I mentioned, this is a symbiosis between, here's our photosynthetic dinoflagellates. These dinoflagellates, when they're in symbiosis, they're actually in a cyst-like form. So many dinoflagellates can form these cysts, or basically just little balls, and they drop their flagella and become kind of amorphic. So all those cool spikes and everything you saw coming off of the dinoflagellates in the previous slide, they lose that and just become these little round balls. And so that's what they look like when they're inside the coral host. They're these brown balls that use sun to photosynthesize and produce their own energy. So they're producing their own sugars, lipids, and proteins for them to grow. But the cool thing about these symbionts is they then either exude or leak or actively transport some of the energy that they make to their coral host. So this image is a little bit grainy, but these are a bunch of little polyps from a coral colony, and I'll show you a better up close picture in a slide or two. But when we talk about coral, they're actually composed of 1 to potentially millions, thousands to millions of these tiny little anemone-like polyps. And so these polyps have tentacles on them, like a sea anemone does. They also have a little mouth on the inside, so they are capable of eating. But unlike anemones, which for the most part, most anemones rely solely on active feeding, most to many symbiotic coral, really most symbiotic coral, relies significantly on their symbionts to produce food that they then give to the host. So without the symbionts, they would die. But the coral isn't the only one benefiting here. The coral themselves provide a home for the symbionts. They provide a safe place for these symbionts to grow and live. And when the coral munch on this food, and it's primarily sugars, but it is some lipids and proteins. When the coral munches on this and eats this food that the symbiont is giving them, they metabolize it just like we metabolize food when we eat it, and they release waste products in the form of nitrogen and phosphorus. And this nitrogen phosphorus then gets sucked back up by the symbionts, the microalgae, and they photosynthesize again with the help of some sun, and the circle just keeps going around and around. And so by nitrogen and phosphorus, this is very similar to the fertilizer you throw on your home yard or garden to fertilize your plants. These single cell plants need that same fertilizer to grow and divide and maintain too. And so it's the symbiosis is heavily dependent on this cycling of nutrients. And it turns out, I think I forgot to include a good picture of it here in this talk, but most coral reefs exist offshore from land, and they exist in regions that are very low in nutrients. And so this symbiosis is critically important in those areas because they effectively trap these nutrients within this symbiosis. And so the coral can suck up a little bit of nutrients that are in the environment, but there's not a whole lot out there to maintain the symbionts at the densities that they actually do. And so it's this recycling of nutrients that is critically important. And so in many ways, for people that may be familiar with, say, tropical rainforests, where the soil of tropical rainforests is actually very nutrient poor, all the nutrients are locked up in the trees and the plants that are above the soil. Within coral reef environments, all the nutrients are locked up within this symbiosis. And so here on the right, I have just one picture. This is a scanning electron micrograph, so high detailed microscopy picture where we sectioned a coral in half. And the only reason I'm really showing you this is all this dense gray material is coral tissue, and then you can see all these little gray balls in here are the symbionts that were living inside this coral tissue. And then down here, I have examples of some coral skeletons. And you can see that so these are dead corals, so there's no tissue on them anymore. If they had tissue, they would have nice, pretty green, brown, purple colors. These skeletons these coral have died or been killed, and then the skeleton's bleached and they produce this nice white skeleton. And again, it's made out of calcium carbonate, just like our bones are made out of. And so the main thing I want to show you here is this is what underneath, so this is a non-living component of the coral that's left behind when they die. And you can also see the diversity of forms and morphology you have to coral colonies. So not all coral colonies are created the same, and I'll show you a picture of living coral in a second that show you that. But you can have quite intricate branching or these brain corals. And even this is actually a solitary coral. And so this would be a single polyp coral. So the mouth would be here, and then tissue with tiny tentacles would cover this whole area too. One little story I wanted, kind of a sidetrack here about the coral skeletons that I like telling the story because it's interesting, but it also is shows how science can be kind of dumb luck accidents at times. Very well known, well respected coral reef biologist from Mexico, Roberto here. He was in Mexico for a long time. He's actually at Penn State now. He's a photophysiologist, so he studies the physiology of coral symbionts. He was in his office, or so the story goes. I wasn't there, but this is how I heard it. He was in his office one day in Mexico, and he had some old coral colonies on a shelf around his office. He was procrastinating or bored, I don't know which, but he had his laser pointer and he picked it up and he was just kind of shining around his office, and then happened to settle it onto a coral colony. And instead of producing a nice, very distinct beam of light like my red pointer is here, he actually saw that the light scattered. And so the nice little, very pinpoint focused laser that he was shooting at it, that point actually got brought bigger. And so this was like an aha moment to him. He's like, what's going on here? And then he realized that what is going on here, and they went on to do some studies and publish numerous papers on this, but these coral skeletons are actually acting to scatter light and magnify it back at the symbionts here for photosynthesis. So not only has this skeleton evolved and provided a rigid structure for this very thin tissue to grow on, but it turns out that it has light harvesting capabilities. Well, not light harvesting, but light scattering capabilities that help increase the photosynthetic input of the symbionts. So light can actually pass through the coral's tissue. A lot of it gets absorbed and used for photosynthesis by the symbiont, but what light that doesn't get absorbed hits the skeleton and then bounces back and gets further absorbed for photosynthesis by the symbionts too. And so what this allows these corals to do is host a larger number of symbionts than may otherwise be possible because this light is scattering. So it's just a really cool evolutionary byproduct of this symbiosis. All right, so I wanted to point out too that a lot of the pictures you have seen and will see show relatively smaller coral colonies, and but these coral colonies, depending on the species and the location, can actually get quite massive. And so this is an example of a Varieties coral in the Pacific Ocean. These divers are drilling down and taking a core of the calcium carbonate skeleton that lies below, because you can use this to learn about the past climate events. But the main reason I'm showing this is to show you how massive this coral colony is. And so, you can see how small the divers are compared to this colony, easily a story or two tall. And some of these colonies can be several hundred to even a couple thousand years old. So these can be quite large and quite long-lived organisms. And some have even argued maybe some of the oldest marine organisms on the planet. So kind of a review of everything that I was just talking about. If we go to a coral reef, this is a picture from Palau in the Western Atlantic, where we did some research while I was a postdoc. But a nice pretty reef with a lot of reef fish. And then you see all these coral colonies, diversity of morphologies that are all around the reef. And so each of these corals is a different species for the most part. And you can see have different morphologies. If we decided to zoom in on one of these coral colonies, like I have boxed in here, we will see that we have these polyps, like I mentioned before. And so hopefully from this image, you can see that these polyps are very sea anemone-like. The difference is that most sea anemones are solitary organisms. Coral, for the most part, except for the solitary one that I mentioned before, are colonial organisms. So they have tens to hundreds, thousands, potentially even millions of polyps all connected. So there's tissue connecting each of these polyps. And so they're one genetically identical organism. So the same genome or genotype on this of a polyp on this side of the colony is genetically identical to this side. And so that they all originated from a single larvae that settled and grew and grew and grew. And so you can see these polyps in this picture are dark brown. If we zoom in closer to a tentacle, you can see there's even more brown. And this dark brown color is actually the symbionts that reside within the tissues, the cells of these coral polyps. And so these, as I alluded to before, the density of these symbionts can be quite large. You can reach hundreds of thousands to even millions of microalgae per centimeter squared, which is about a ½ inch squared. So a very tiny portion of the colony can hold millions of cells. So you can imagine that in a colony this size, you could be topping out in billions of cells. And so each coral colony can almost be thought of as a phytoplankton or a microalgal bloom because there's so many cells here. So the reef itself, I wanted to mention too. So you can see we have coral colonies growing here. As they grow and get bigger, they're laying down successive layers of calcium carbonate, the skeletons I was showing you before. And that's really what makes a coral reef a reef. Without the coral, it would be basically a sandy bottom with very little to no three-dimensional structure. But these coral, as they grow and get bigger, and as they die and new ones settle and grow on top of them, they create all these nooks and crannies and large three-dimensional structure that provide the homes for all the reef fish species that we see, and of a lot of other invertebrates like this crinoid you see sticking its arms out here. And so it's these, if it weren't for coral and this Microalgual Symbioses, this reef would not exist, and the species that depend on this reef would not exist either. So they're extremely important to this ecosystem, and as I'll talk about here, are extremely important globally too. So the top two images I just wanted to show that this is still in Palau when we're doing some work. This is an offshore reef. This is a more inshore reef. You can see that different reefs have different structures. You can see that different reefs have different morphologies of coral colonies growing. And so not all reefs are the same. And but they're highly diverse in both coral species and the marine species, fish and invertebrate that live on this reef. When we talk about coral reefs and where they're found, they're primarily found flanking the equator. So the equator would be somewhere around here, and you can see all these little dots. You can ignore the colors for now. All these little dots are locations of known shallow water coral reefs. And You can see they flank the equator and roughly lie between the Tropic of Capricorn and the Tropic of Cancer. So 23 degrees north and south of the equator. So these are definitely a warm water ecosystem. As you move up the east coast of the United States, for example, there are some corals that go all the way up into Maine, even symbiotic corals like you find here, but they don't form reefs like you see in the tropics. The colors here, which I'm not going to talk too much about here, this is an old map now, 2011, so that you probably see a lot more reds and yellows, but they're classifying reefs according to their local threat level. And so reefs, to make the point that reefs are not only threatened by a changing climate, particularly warming seawaters and potentially acidifying seawaters from CO2, but also locally, reefs are threatened by stressors such as overfishing, coastal development, which can lead to pollution and sedimentation. You have aquarium trade that has in the past and still does in some places, where they use things like cyanide and dynamite to capture organisms for aquarium trade. All these things can stress and even kill a reef. And so, but by far globally, the largest stress to coral reefs is a warming climate. And so these systems are incredibly important to the coast, to the oceans worldwide, and have mentioned down here that they support approximately a quarter of all marine species in some form or fashion. So in some cases, some species live their entire life on a reef, but others may use them for feeding or reproduction. And so the juveniles may live there until they're big enough to venture out further. But it's estimated that a quarter of all marine species are dependent on coral reefs. So as we lose coral reefs, we lose the benefits that those reefs provide. And they're also, and this number is probably low by now too, is much higher. Over 500 million people depend on coral reefs for food or economic purposes. And they also provide coastal protection to storms, just like salt marshes do in, say, the Port Royal Sound. They dampen wave energy, and when storms come ashore, they prevent or help prevent erosion to the terrestrial environment. What's really crazy about, as important as these systems are, they cover somewhere I have 0.1%, maybe a little bit higher, but definitely less than 1% of the ocean seafloor is covered by coral reefs. So hugely important, but not very abundant by an area basis. And so they, as we continue to lose coral reefs and they don't come back very quickly, we're losing all the benefits that they provide. And so I'm beginning to ramble a little bit, so I'm gonna try to speed up a little bit, but I mentioned that temperature is the main thing affecting coral reefs on a global scale negatively. And so I'm sure you've all heard of mass bleaching events. And so what bleaching is this used to be a nice brown colored colony filled with symbionts, but now temperatures were too extreme, too high for too long, and they spit out the symbionts, or the symbionts leave. Again, there's debate on that, but the symbiosis breaks down, the coral bleaches and turns white, because what you're seeing below it now is you're seeing through the coral tissue and you're seeing that calcium carbonate skeleton. And so it's called coral bleaching because they turn white. And it turns out that coral bleaching is reversible. So you can, this little pictorial diagram here is showing that you can have a healthy coral. If it's stressed by water temperature for days to weeks, it can become bleached, so it spits out the symbionts here. But if conditions return to normal, so water temperatures decrease, then over the coming weeks to month, it can actually brown back up, those symbionts come back, and it can return to a quote unquote healthy state if given enough time. However, if the stress event is prolonged enough, so if it's either too hot for too long or just too hot too quickly, it will eventually kill the coral because the symbionts don't come back. The coral essentially starves to death and they die. And so you lose that coral, and you can potentially lose an entire reef system if bleaching is bad enough. And an interesting phenomenon that was discovered fairly early on with bleaching events was that not all coral colonies or even sections of coral colonies bleach the same due to the same stress event. And so here, this phenomenon is known as differential bleaching. And you can see that you can have, it may be hard to see, but here's a bleached coral here. This is in La Paz, Mexico, in the Eastern Pacific. And then here's healthy coral down here. This is actually the same species of coral, but one hosts one sensitive symbiont. This one hosts a stress tolerant symbiont, and so it doesn't bleach. Here we see different host species that don't bleach, and here we see a coral colony where parts of the colony bleach and other parts don't. And so what all this is showing is that depending on the host and the symbiont species that is being stressed, some can weather the storm better than others. And so I am going to try to speed up a little bit to get towards some of the data I have. But a question early on was that when you isolate these symbionts in the host, you can take them out of the tissue with basically a water pick, like you use to clean your teeth. You can water pick coral tissue off the skeleton, isolate the algae, and look at them under a scope. They all look basically the same, except some are smaller or bigger than each other. And so originally they were thought they were the same species worldwide. But through the 70s and 80s, it was quickly discovered through a variety of experiments that they have distinct behavior. And this was mostly with cultured. So you can take these algae out and culture them in the lab without the coral. But through behavioral morphology, specificity, physiology, even their biochemistry, and even chromosome number showed that there was more than just one species of symbiont. And so today we know that there are numerous species of symbionts, and they all show very clear ecological patterns according to species. And so we see very high specificity between particular coral species and symbiont species within a given environment or a geographic range. We see particular symbiont species that are adapted to different environments. So some are better adapted to high light, some are low light, some are better adapted to stress like temperature changes, some are more sensitive to that. Some appear closely associated with the type of symbiont transmission method. And so in the coral world, they produce these little larvae that are formed from sperm and egg coming together. Most coral actually spawn their sperm and egg into the environment, and the larvae are formed outside the colony. But there are a small subset of coral that actually internally fertilize, and these that internally fertilize will pass their symbionts from one generation to the next, whereas these will acquire their symbionts from the environment. And then, of course, with this diversity, we're seeing that particular species have very distinct geographic ranges, and that really there's really only a few species that seem to have a worldwide distribution, but most are limited to a particular ocean basin and even a region within that ocean basin. And so when I first started my PhD dissertation, the genus was called Symbiodinium. Genetically, we could show that there was huge diversity within this genus. And even the first paper back in 1992 noted that at the time, these were called clades, which clade is just a genetic grouping of like individuals that are genetically similar to each other. And the original paper, that first gene sequences for Symbiodinium, showed that the diversity within this genus is comparable to orders for other non-symbiotic dinoflagellates. But things don't always work very quickly in science, and so you have to build data over years and years to convince people what is occurring. And we now know, and it only took about 25 years, that the genus Symbiodinium is actually more like a family, if you're familiar with the biological classification system. And so each of these clades is now a different genus, and this just occurred a few years ago, back in 2018. But I like telling this story to my students I'm teaching. You know, we talk about, everybody wants to know everything about coronavirus and COVID, for example. Why don't we know more? Here's a good example of how slow science can be at time. And so the amount of progress we made on diseases like COVID is quite amazing compared to other areas of scientific research. But just to drive the point home, here's, again, one of these family trees, a phylogeny of our different diversity groupings within the symbiont family, Symbiodinaceae. You can think of these, and each of these is going to have numerous species within them, is something like that you may be more familiar with, like the cetacean families, so the whales and dolphins. And so you can imagine that something like a narwhal has a very different physiology and behavior and geographic range than something like the bottlenose dolphins that we have in the low country. And so the same thing is true for species within and between different genera within the symbiont group of organisms. So there's a large amount of diversity within this group and a large amount of physiological diversity too. And so I will try to quickly go through some data at the end. Chris, feel free to tell me to stop talking at any point. But just a little bit, I'm not going to go into too much technical detail, but some data on, that I generated during my PhD dissertation. By the time I started my PhD, we knew that there was high specificity between coral species and symbionts. And by that, I mean that not any coral species can form an association with just any species of symbiont. They recognize each other and they form very distinct symbioses. And that's particularly for a given geographic range and environment. But the questions became, well, how flexible and stable are these associations? Given that changing climate is causing coral bleaching, and these systems are being stressed beyond what they can handle, we're having massive reef die-offs. Is it possible that new symbiotic associations, new species can recombine and kind of form stress-hardier supercorals to help save reefs, essentially? And so that's where my research really came in. Up to that point, most genetic research was being done sequencing, looking at different species. But I was, and a few others along with me, were getting to the forefront at looking at within species diversity. So looking at populations within a species from different geographic locations, and even diversity within a population. And that'll make more sense here in 1 second. I don't want to go into too much detail with microsatellites, but these are the markers, genetic markers that I was using at the time. These are basically, if you remember, DNA has your ACTs and Gs, the nucleotides that make up the genetic code. Microsatellites are areas where you have repeated motifs, or things like CA, 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 GT, GT, or GTT, GTT, repeated. And for reasons that, if anybody's interested, I can talk about later, these regions mutate at a high rate. So when you're sequencing a gene, point mutations in the genetic code are on the order of 100,000s to millions of years time scale. These, the mutation of these markers is on the thousands to tens of thousands. So you can look at very near-term evolutionary questions. And you also can find them throughout the genome. So you can not just sequence one gene, but you can essentially categorize numerous loci within the genome of this organism. And so when you do that, if you use multiple locations, you can generate a multi-locus genotype or genotype, or effectively genetically identify an individual within a population. And this is essentially what we use when we do forensics genotyping of humans, say from crime scenes. You may look at these two individuals. They are both humans. They both look similar, have dark hair, mustaches. And they don't look a whole lot different, except they're different shape and size to them, kind of like Symbiodinium are when they're inside their host. But if you genetically characterize numerous microsatellite loci within these individuals, you can show that this individual is genetically distinct and different from this individual, correct? And so we can do the same thing with both the symbiont that reside inside the coral and the coral themselves. And so we're using this kind of forensic type technique to look at individual diversity. And so these colonies may look the same and they both appear kind of brown or green color, but in fact, the coral that hosts the symbionts is genetically distinct from each other, and the symbionts within those colonies are different too. And so we first, this may look a little bit confusing, but these are basically two statistical methods at clustering individuals. And so first, one of the things we started doing was based on gene sequences, we thought these were distinct species. We used population genetic markers, the microsatellite loci on them, and taking the data, we can cluster them. The ones that are more closely related have similar genotypes, will cluster closely together, and we showed that different species are species clustered with themselves and not others. So the different colors and shapes are just indicative of different species, and you don't really see any overlap between these clusters, indicating that these are distinct species and they're not reproducing with each other. So I forgot to mention microalgae they can reproduce asexually, so they make clonal copies of themselves, and coral can do this too. But microalgae can also sexually recombine, so they can come together and create recombined and new genetically distinct individuals, just like humans do with a sperm and egg. And so these populations were showing that they're not sharing genetic material, so they're distinct species. And then this is the main story of the talk I wanted to quickly talk about. This was work we did in the Eastern Pacific, primarily in the Gulf of California near La Paz, but we also sampled 5 other locations in the Eastern Pacific, 2 in Mexico, 1 in Panama, Galapagos, and the Clipperton Atoll of this coral here. This is a Pocillopora species in the Eastern Pacific. These reefs are really unique and different from just about all of the reefs around the globe in that these reefs are monomorphic. This is the only species you really find on the reef. It dominates the reef. You go down underwater and dive. And for those that have been on coral reefs and see the diversity of coral, or even from the pictures I was just showing you, this is basically just a carpet of Pocillopora just kind of rolling through the ocean. And so it makes a very cool model system, very neat model system to ask some questions. And what's neat about this system is it's the same coral species, but it can host 2 different symbiont species. One is stress tolerant, this D. glynni, and the other is a stress sensitive one. This one hasn't been named yet, but it's C1. And so there doesn't, as far as we know, we can't find any genetic reasons why they host one over the other. But what happens is when there are stress events, the ones that host stress tolerant symbionts survive. The ones that host C will bleach and may eventually die if it's severe enough. And so we came to study specifically this symbiont here with regard to questions concerning climate change. And we wanted to look into the evolution of Symbioses, where we can control, for the most part, the host, but we have 2 different symbionts and kind of see what happens. I wanted to mention too, and this will become important as I get to the results in 1 second. These are maternally inherited, so the mama corals give their symbionts to the baby larvae as they leave the colony. And so just quickly, we did random transects and polar plots where we actually tagged colonies. And so we were able to look at the same colonies over multiple years and look at their symbionts and their the genotypes of the symbionts. And this is an example from La Paz, but every location I showed you on the previous map had anywhere from 3 to 5 reefs that we sampled that were separated by several kilometers or more, just to increase our sampling size and potential for diversity. One thing we discovered when we were doing this, and there was another paper out a little while before we did this that suggested this was the case, is when we sampled colonies multiple times, we found that one individual, one clonal genotype, either occupy the entire colony or a majority of it. And so what we found is that colonies were limited in the number of genotypes they could find in those in that colony. Remember, this is the same species. We're looking at the stress tolerant D. glynni here. They were either dominated by one or two genotypes, and when they are dominated by multiple ones, they tend to not be mixtures, but dominated certain sides of the colony. So that was really interesting. You know, instead of being 5, 10, hundreds of different genotypes in here, which would, from an evolutionary standpoint, might give you the ability to create your symbiosis, it was very stable at even the clone level. And so what this means is that if you look at, you know, this is not in the Eastern Pacific, but this is back in Palau again, where you have a huge diversity of coral species and colonies here, that these coral colonies are kind of acting like culture vessels for the symbionts. For those of us that are interested in studying these symbionts, you can go to a coral colony, and if you have a little bit of background information, you probably know what species it is, and you can be fairly certain that it's only a single genotype of that species. So you can start and begin to ask some really interesting questions on the diversity, the physiology within a species of these symbionts. One interesting thing we found, remember these symbionts are maternally inherited, so the mama colony gives the symbionts from its tissues to the larvae. When we compared the genotypes for both the symbiont and the coral, they did not match. So you would think that a coral that is giving its symbionts the next generation would form these maternal lineages of symbionts, but we didn't find that. And so what these are just two examples of plots from the Gulf of California. The color is the symbiont genotype, so green are the same genotypes. The shape is the host, so the squares are the same. Square and circle will be two different genotypes. And what you see is, and statistically we showed that at the species level, so a particular species of coral with a particular species of symbiont, you can have host-symbiont recombination. So remember, this was a question we were asking concerning the evolution of coral algal symbioses with regards to a changing climate. And so it appears that at the species level, you can swap genotype for genotypes. And what was really, really fascinating and interesting that we weren't expecting, when we looked at our populations, we did statistical analysis between the samples we collected in the greater Eastern Pacific with those collected in the Gulf of California. We found that this population to the south is distinct and rarely shares genetic material with the population in the Gulf of California. That's not too surprising. You can ignore this up to the right and ignore this for a second. I'll describe in a second. It's not too surprising because the Gulf of California, there's a biogeographic break here where you find endemic species within the Gulf of California that you don't find in the Eastern Pacific or even on the other side of Baja California. So that wasn't too surprising. What was surprising was when we looked at the host genetics. So remember, we have the genotypes for the host. This is that same cluster analysis I was showing you with symbiont species before, but now we're looking within a single species. And these site designations here are going from north to south. We don't see a distinct break. So Gulf of California is here, then all of these are your southern sites. We don't see a distinct break in population, in the populations within this host. And in fact, we see that genetic material is frequently shared between this biogeographic break, at least frequently enough to maintain a single population. The yellow you see creeping in down here is a pattern in population genetics you see. It's called isolation by distance, where instead of seeing a distinct break between populations, you see that this population and the ones to the further south, the most distant populations, are beginning to diverge just because of distance alone, not because of other environmental or physical parameters. And so what's crazy is that the hosts are going back and forth. They're carrying their symbionts with them, right? because they're maternally inherited. But when they get to a new location, whether they're migrating in or migrating out, they don't maintain the same symbionts they came with. And so it appears that in this example, we're hypothesizing larvae are traveling from the outside in, but it could go the other way potentially. That the orange southern population carries its symbionts with it. It settles, but upon settling, it takes up local symbiont population from the Gulf of California, and that replaces it within that colony and within that larvae. And so we thought this was very exciting and strange, because within this system, you expect there to be fidelity among host and symbiont genotypes. But if you remember back to the example I showed you in the Gulf of California between two reefs, within a single reef, we saw that there was genotype switching between hosts, between the coral and the symbiont. So we actually already had some evidence that this was possible. And so when we look at the environmental conditions between the southern locations in the Gulf of California, what you really see is that starting right around this break here, you're really moving from a tropical environment to a subtropical environment. And so what I'm showing here is sea surface temperature average over the course of the year for, this was about 10 years, I believe, if I remember correctly. Oh, sorry, yeah, 2000, 2009, sea surface temperature, and then PAR is a fancy word for light. And the red is the Gulf of California population. The other symbols are all the southern populations. That it's not that we necessarily see a colder versus a warmer population, or a higher light versus a lower light population, which we do see some of that, the extremes, but what we really see is high variability. And so what we think, and that's what this graph is showing here, is the same data, it's just showing the variation over the course of a year, that this population in the Gulf of California is much more subtropical. Tropical populations or tropical regions tend to be much more stable in their environmental conditions on a yearly basis. Subtropical, and as you move further and further north, the variation becomes larger and larger, right? So as the East Coast, as you move further up the East Coast, you get into regions where you can find snow and cold winters, things that we don't have down in the low country around Beaufort County. And so the same thing is occurring here. And so what we think is going on is that we have an example of local adaptation by this population. And when new individuals come in or out, they are outcompeted by individuals that are better adapted to the local conditions. And so this was really neat, because this had not really been, it had never been shown before for coral algal symbiosis. And so this local adaptation and this switching between genotype and genotypes, which is would be a direct way for these symbioses to evolve and adapt to a changing climate. I am going to unfortunately skip this because we're running out of time. I do apologize. If anyone wants to hang around a little bit longer and hear a cool story about an introduced species of symbiont that we found in the Caribbean, I'd be happy to talk about it. But so what, just from the example I just gave you with the Possipore in the Pacific, we can come back to this question: how flexible and stable are these associations? It turns out that they can be quite flexible, but at the time when we were doing this work, it was historically focused at the species level. So can a particular coral species take up a new symbiont species? And that's what this little graphic is showing here. Here's a coral dominated by one or a couple species. There's a selective sweep or a disturbance in the environment that causes it to shift towards another species. Can we get new symbioses form at the species level that will help these symbioses evolve? And while I am not saying that this is not possible, clearly these species combinations, recombinations occur in geological history. Genetics show that it happens, but probably not on a very frequent basis, and not frequently enough to save all the species of coral and coral symbiosis that exist. And what we argued is that really a more appropriate way to look at coral algal evolution and its ability to adapt to the changes in climate that we're seeing, it's really going to happen at this individual level, where an individual genotype of a coral colony that happens to be more resistant to whatever the stress is makes or forms a combination with this green individual, not species, but individual genotype that is also more stress tolerant. And this super symbiosis occurs and can potentially face the problems that are mounting towards coral symbiosis and coral reefs in general. And so it's a very gradual change, as Darwin had suggested way back when. I just want to very quickly, and then I promise I will stop, wrap this back around to what in the heck I'm doing in Beaufort County at USCB and working on water quality and phytoplankton in the Port Royal Sound. So those are symbiotic microalgae that, want me to stop?
No, I don't want you to stop. I just wanted to let you know that there is no rush and you can absolutely take your time and explain everything. So don't worry.
Thank you. Yeah, I apologize. I tend to ramble. But so we were talking about microalgae and symbiosis with the coral reefs. The work that I am doing and beginning down here in Beaufort and the Port Royal Sound is really focusing on microalgae that are commonly called phytoplankton. So these are the microalgae that exist within the water column. They only make up about 2% of global biomass, but they are the base of the food chains in oceans, estuaries, and even freshwater lakes, rivers, streams. And so that's what this image is showing here, is these phytoplankton are the base of the food web that everything depends on. These are the organisms that take sunlight and nutrients in the water column and multiply and divide and provide the food for everything above it. And not to mention, they also produce a large amount of the planet's oxygen, which is important to us, but particularly for aquatic species that depend on that oxygen to breathe and survive. And so this is what I am doing now in the Port Royal Sound, is characterizing our environmental conditions around the sound. We have some baseline knowledge that has been collected over the years through DHEC and DNR state agencies and even NOAA. But we're trying to collect that on a more frequent basis to get a better idea that it is a tidal system down here. And so tidally things can change 4 times a day as the tides go in and out. And on top of all that, we have a county that is quickly growing. And so again, this is an old slide that I used when I first moved here, but as everybody knows, Beaufort County, particularly Bluffton, but Beaufort north of the Broad is potentially quickly following, is rapidly growing. And so as we alter our land use and change forested areas into essentially urban and city type environments, what goes on land is going to affect the water quality that we see in our wetlands and estuaries around the county. And so the dream that I have coming down here is to establish a long-term monitoring system of numerous sites around the Port Royal Sound watershed. And when I say Port Royal Sound watershed, I do include May River, Calabogie Sound as part of that. Where we monitor water quality conditions on a weekly basis. And so since 2002, my lab and I have been doing 3 sites, 1 on each of the main stems for the Sound, the Beaufort, the Broad, and the Chessie. These red dots are proposed locations. As many of you know, we've started a citizens monitoring network in collaboration with the Maritime Center and the Port Royal Sound Foundation. And so individuals and groups are beginning to collect samples at locations like these. And in the near future, we'll be bringing some of these sites online for weekly monitoring, just like I do on these 3 sites. And at these sites, we measure environmental conditions that are important for biological activity in the sound, like temperature, salinity, dissolved oxygen. We do that in the field. And then we bring back samples to the lab to look at chlorophyll concentration, which is a quick and dirty way to look at phytoplankton or microalgae biomass in the system, nutrients, which are going to fuel this phytoplankton, total suspended solids, which are going to give you an idea of the light field that these phytoplankton experience and how that affects their photosynthesis. And then we're preserving samples to eventually start doing genetic analysis on the phytoplankton and bacterial communities that exist in the system. And so I should have a Port Royal Sound graphic here, because they're together, we're putting together the citizens monitoring program that is going to allow my lab to analyze samples from this vast area, because we can't sample all these locations, but you as citizens can. And I'm very excited for this to get up and running. And lastly, and then I promise I will stop, I just wanted to announce a new funding situation that occurred back in May. I received a nice amount of funding from the Community Foundation of Low Country to establish a continuous monitoring station at the Maritime Center on their dock at the on the Chessie River. This is not their dock, obviously, but this is what the system will look like. It's going to contain both sensors to monitor water quality, temperature, salinity, dissolved oxygen, and chlorophyll, and turbidity, and also a weather station on the dock. And it will be able to collect data as frequently as every 15 minutes, but most likely on an hourly time basis. And so this data is data that I had generated from projects I did back in Delaware using the same sensor setup, but we'll be able to get this really high resolution change in environmental conditions and describe our system even better, and hopefully be able to use this data to both look for change in the environment, which may indicate problems in the watershed somewhere, and key us into stuff we need to look into further, but also use to predict out into the future how things may change as the lands around the county change, but also as our climate continues to change too. And we're doing all this not only with help of citizens, but undergraduate researchers at USCB. This is an old picture from the previous summer, but we have about 9 students working in the lab this summer on various topics concerning water quality in the Port Royal Sound watershed. And so lastly, I'll finally stop and ask you if you have any questions.
Yeah, I got a question. I got lots of questions. So one, where did you find that picture?
It's floating around on the web somewhere.
Beautiful.
I even got my toenails painted on that one. That's a gem.
I thought you
yeah, I really enjoyed that. Thank you. I have all the questions. If anybody wants to type in any questions, please, you can do so right now. I will just go ahead and say thank you, Ty, very much for that presentation. I thought that was really nice. And hopefully I'll have it posted on our YouTube channel in the days to come. But let's talk about some of my questions. So I don't know if you had mentioned this or not, with the coral symbionts, when they leave, expel, they're able to retake them back in.
Yeah.
And I don't know if you said this or not, or if I missed it. Are they able to, do they take in different symbionts then in the reuptake?
So yeah, let me actually, I'll go back to this slide, which I didn't really talk about. But this graphic kind of describes this, that when we look, in this case, we're not talking about genotypes with these colors. These are different species, for example. And so you can, and this is a bleaching event that happened in Barbados in the Caribbean, coral bleached, the ones that had this red stress tolerant symbiont. Weird things are going on with this system, but the ones that had the stress tolerant symbiont didn't bleach as badly. The other ones, the ones that didn't, did, and some died. But then as they recovered, they recovered back to the dominant species they had previously. And so what I'm trying to say in a long-winded version to your answer is that they can take up different symbionts, but what typically and has happened, at least from what the data shows, is that the ones that do survive a bleaching event, they revert back to a symbiont species that they had previously or could have had in that same environment, right? So I simplified it a little bit, some depending on the coral species, or even like the La Paz, the Eastern Pacific example I gave you, the Pasipora there can have C or D, or the two different species. The same thing is true for other coral species in other parts of the world. They may associate not just one species in a particular geographic region and environment, but two or three species possibly. And so, but they tend to revert back to the ones that they had before, showing high specificity. So, and that's where a lot of the questions were coming from before was, is a bleaching a mechanism for change? And so that new things can come in and create new symbioses. There's some evidence to show that it can happen, but the frequency at which it happens is at least to my opinion, questionable.
Yeah. So does each coral species then have a preference to the symbionts, or is it just kind of random per individual?
No, very specific. And so I think some groups are slowly getting close to figuring out what that is, but there is some kind of like lock and key mechanism. They recognize each other.
Right.
And so that's where with some background sampling and a lot of certain parts of the ocean have been highly characterized now, that if you go to a particular location at a particular depth and sample a particular coral species, you can narrow it down to probably 1 to 3 symbionts that you can find there. But if you sample the reef as a whole, there could be 50 to 100 different species on that reef, depending on the diversity of coral on that reef. But okay. Does that make sense? So like,
yeah. I just find it's just like really hard to wrap your head around, right? That there is a lot of, I guess, they can have diversity in them, but they have a preference, and yes, yeah, that's just interesting.
Yep.
Cool. You were talking about age of corals,
yes.
And how large corals can be, right? Just for whoever views this later, what is the oldest coral, or what they estimate to be?
Funny enough, I was kind of looking at this while I was getting this talk ready. Most of the ones I'd heard about, well, so symbiotic, so tropical corals like we're seeing here and I talked about, they were, most limitations that put them around 1,000 years at the oldest. But there have been newer discoveries and using genetics and like sampling a massive coral like that big one I showed you before, sampling it on one side of the colony and then going all the way to the other side, looking at genetic. So if something's 1,000 years old, they're likely to build up mutations within individual cells. And so through modeling, you can kind of get at what the age that colony is. And so some newer studies are putting it more at maybe they could be up to 4,000 to 5,000 years old.
Oh, wow.
And deep sea coral, that's actually very, it's not uncommon for some species to live thousands of years. Tropical species weren't necessarily thought to live that old previously.
Yeah.
But
yeah, I would like even question like how long they had, if they were even there 1,000 years ago, just with sea level coming and going, what that had looked like. But I wonder too, like, do you see genetically identical corals on the same coral reef in different spaces? Because I know like with what's it called, fracking, where they take the fragmenting, like does that occur naturally too on the reef?
Yeah, and so that's what I kind of glossed over with this. So the coral that fragment the best are the branching species because they're more fragile.
Right.
And so like this ossipore that we were studying in the Eastern Pacific, it fragments very frequently. And so you can imagine that a big wave comes along, a fish, a human, whatever, a storm, it breaks off a piece, it falls to the ground. If it's a suitable location, it can grow a whole new colony. And so the data that we had here, that's what this is actually showing. These shapes, like this circle, at least with the resolution of the genetic markers we have, this is all the same individual. So it'd be like having 10 Chris's hanging around your office. You have 10 of this coral genotype. And what's really crazy, I took it out of this talk, but there's one reef that was more of an impacted reef, but we had 3 of these transects along about a 100 meter distance along that, so 300 feet or so. And one single genotype dominated something like 80% of the samples was the same genotype. And so that genotype itself is likely relatively old too, just from the fact that it could fragment and then grow new colonies and disperse. So, but not all coral really fragment that. You can physically fragment corals yourself,
right?
Even the ones that don't normally fragment, like in the aquarium trade, you can cut them up into smaller pieces and they'll grow new colonies. But it's really not all coral colonies in the environment fragment to.
Right. Well, that was, I guess that question stemmed from the age. If you're testing fragmented corals, does that throw off the dating process of like how old that species actually is?
It could be, and I would argue that it could mean that it could underestimate things, because you could have the original colony that no longer exists, but its fragmented offspring still do. So its clones are still hanging around, but. So yeah, and that also, it brings up interesting questions again from a adaptation evolution standpoint. How much diversity is out there and how much diversity do you need on a particular reef for a particular species to persist as the environment changes, right? Some of these may have been around for hundreds of thousands of years, and they've lasted this long, but they also haven't seen things change necessarily to the degree they are now.
Sure. Cool. Well, you answered all my questions. I really appreciate it. That was a great talk. Thank you for taking the time out of your day participating.
Sure. Of course. Sorry again for rambling a little bit.
It's funny is this is being recorded, so I'm going to first say that I am very fond of Ty, so I do like picking on him. I'm sure he feels the same way, as you could see by his final picture on his slideshow. But yesterday we were talking about how long does this need to be, and you said 30, 45 minutes. And I thought to myself, I'm like, never in my life have I had a PhD last 30 or 45 minutes on a talk. I was like, we'll see about that. Here we are, hour and a half later, finally over.
I'm glad I could reinforce your preconceived notions of us.
Next time I will definitely make a hard stop. I will tell you, hour. That's it. That's all you have. Well, again, thank you, Ty. You can now leave Clemson and go back and hang out with your family. I hope you have a great vacation.
Yep, thank you.
And I'll talk to you next week.
All right, sounds good. Thanks, Chris.
Right. See ya. Bye.